Hi, everybody. Welcome. My name is Sophia Kim. I'm the director of the Science Learning Institute, and we're here for our first STEM equity seminars for OWLs for the school year. So we're really excited to have people here and people on Zoom. Before we get started today, next slide, we want to read a land acknowledgement. This is a land acknowledgement that Foothill College has created to just acknowledge the land that we sit on. So I'm going to read it out. For Shade Truhi. Foothill College recognizes that it is located on the ethno-historic territory of the ancestral and traditional land of the Puichon Tamien Ohlone-speaking people, and the successors of the historic sovereign Verona Band of Alameda County, presently identified as the Muwekma Ohlone tribe of the San Francisco Bay Area. This land was and continues to be of great importance to the Muwekma Ohlone tribe and other familial descendants of the Verona Band. We recognize that every member of the Greater Foothill College Los Altos Hills community has and continues to benefit from the use and occupation of this land since the institution's founding in 1957. Consistent with our values of community, inclusion, and diversity, we have a responsibility to acknowledge and make known through various enterprises Foothill College's relationship to Native peoples. As members of the Foothill College Los Altos Hills community, it is vitally important that we not only recognize the history of the land on which we live, work, and learn, but also we recognize the previously federally recognized Muwekma Ohlone tribal people are alive and flourishing members of the Foothill College Los Altos Hills and broader Bay Area communities. Aho. So thank you all for hearing that one. So today we have Dr. Jennifer Ortiz Cárdenas from Stanford. I'm just going to read, well, I just shut. I'm going to read a little her short bio. Jennifer and I met, I guess, this past spring or so, partnering on a field trip that we were able to do together this summer, which I think some students I know here were at. So Dr. Jennifer Ortiz Cárdenas is the director and outreach coordinator of the Microfluidics Foundry at Stanford University. She received her BA in chemistry from Binghamton University and earned her Ph.D. in chemistry at the University of Virginia under the supervision of Dr. Rebecca R. Pompano. In her research, she developed biomimetic photo pattern 3D cultures to recreate the spatial organization of tissue on microfluidic chips. In her current role, she is in charge of the Microfluidics Foundry and supports nearly 30 users in integrating microfluidics in their research. Additionally, she is passionate about improving diversity and inclusion in STEM and has developed outreach programming to increase the representation of Latinx and other URMs in the microfluidics field. Today, Dr. Cardenas' talk is titled Microfluidics as a Vehicle to Increase Equity, Diversity, and Inclusion in STEM. So just really excited to have you here today. So thank you for joining us.
Thank you. Thank you for the introduction and also for the invitation. Let me try to share my screen. That will work.
Yes.
That's a little blurry.
We go to the next slide and see. It looks clear on the Zoom. So for the audience at home, at least they can see.
Sorry, everybody that's here. Yeah, so again, thank you so much for the invitation. I'm really excited to be partnering with Foothill, and I really wanted to take any opportunity I can to come up here and talk to you guys. And it's really nice to see some familiar faces again. So today I'm going to be giving you a talk. So the title of the talk is what I'm currently doing. But in the talk, I'm going to talk about my path in the academic sphere and how did I end up where I am today. So just like the outline of the presentation, I'm going to give you a background of like where I come from, where I grew up, and then experiences in undergrad, research experiences, my graduate school work, and then the current work that I'm doing. And before I begin, I wanted to say that even making this presentation, I've never given a presentation talking about my path, and it was very nice to reflect back and, you know, think about all these crazy memories that I have. I'm very excited to share with you. So I wanted to start with a picture. It looks prettier on Zoom. I sorry, of the town where I grew up in. I grew up in a town called Luquillo, Puerto Rico. It's on the east side of the island, and it's a seaside town that is very popular amongst locals and tourists for surfing. So this particular spot, it's called La Pared. And my house where I grew up was probably like a 4-minute walk to that beach. And I grew up living, like I'm an only child, and I grew up living with my parents and my grandma in a house, and I'll show you a picture of that. Why is this moving without me moving it? Okay, so this is a picture of like, oh my God, it looks so bad in the screen. It's a picture of a very famous spot in my town that I love to go to. It's called the Kioscos, and it's a row of like 60 individual kiosks that sell like fritters, piña coladas, drinks. So you can go to the beach and then go to the kiosk to like get some food and then go back to the beach. And this is where I would hang out like when I was a teenager. And I didn't used to go that often, but this is where I would go whenever I left my house. So this is a picture of the house that I grew up in. And that's me with my dad. And as you can see, it was a very humble house. I grew up in a low middle class family, and my parents actually, when I was small, their job was to go around different towns on the island and try to sell people encyclopedias, if you can remember those. And then eventually, when computers began to be a thing, they would go and try to sell computers to people. So I grew up around a lot of books, a lot of, my mom really instilled in me that studying was a way to achieve whatever I wanted. And so I took that to heart, and I was like really into school. As you can remember, I said I was an only child, and like one of my favorite things was to line up all my dolls and pretend that I was a teacher. And I didn't have a blackboard in my house, so I just used the back of the door for my room. And here I am giving a lesson to my dolls. And I was, very motivated to becoming a doctor one day. And I did end up becoming a doctor, just a different type of doctor. And I'll talk to you about like how that change ended up happening. So another thing is that I lived in this town until my 10th grade in high school. And I went to public schools like my whole life. So whatever the closest school to my house was, that's where I went. And when I got to, when I got to at high school, my biology teacher saw that I was really excited about his class and that I was like paying a lot of attention in biology. And he told me, I know of this school that specializes in science and math, and you should apply and you would be a great fit. The only problem, it was that it was all across the island on the other corner. And so it was a boarding school, and you could stay there for free because it was also a public school. So you could go and stay there and study for the weekdays and then go back on the weekends. So my parents did the sacrifice of, once I was accepted, they would drive me each Friday or each Sunday to drop me off for school, and then they would pick me up on Fridays. And a lot of people, this was a school that you had to apply to get in, so you had to pass this standardized test. And at that time, I always hated standardized tests, and I was freaking out thinking that I was never gonna get in, but I did get in, and then we had to figure out how to get there and back. And it was really nice because it was a lot of people from all over Puerto Rico that would go to this school, and I met a lot of my really good friends that I still talk to this day. And this was a time in my life where I felt really challenged by science and by math. And by the way, I never really liked math, but here I am. This is a picture of the, oh my God, so blurry, of the school. It was by no means glamorous. It was still a public school, but the classes were really interesting. And, I took classes like anatomy and intro to biochemistry and things like that. And so based on these experiences, I decided that I wanted to continue to challenge myself. So when I was applying to go to university, I applied to the schools in Puerto Rico. But then I was like, oh, maybe I could go to United States. And at the time, my mom said, okay, if you want to, you can try, but we don't have a lot of money to pay for a lot of applications. And at the time, I think they're better advertised now, but I didn't know about the waivers for admissions and things like that. So my mom just told me, pick one school, and if you get in, then you can go. So that was a lot of pressure because even though I had all these experiences where I was taking really hard science and math classes and I was doing well, I didn't want to have my mom like lose that money. So I was very strategic on where I was going to apply. And I thought there's probably like a really low chance that I'll get into like a fancy private school or that I could even afford if I got in. So when I was, should I stop this?
Is it on automatic or is it?
1 second. Maybe it's this.
Oh.
Okay, so when I was applying to school, I really was thinking, okay, maybe I can go to a public school where the tuition won't be as high. And then if I get some scholarships, like that would get me close enough to the amount that I would have to pay for college. And I always wanted to go to New York, had never been to New York, but I was like, New York sounds pretty fun. So I looked up like, what are good schools in New York that are public and have really strong science programs? Because as I mentioned earlier, I always knew that I wanted to be a doctor. So my hope was to go into a pre-med track. I really enjoyed chemistry in high school, so I wanted to do that. So I ended up applying to Binghamton. I ended up applying to Binghamton and I got in. And so at this time, I was very excited because, you know, I've always stayed in Puerto Rico, had never gone anywhere. And I was also nervous because up to this point, my entire education had been in Spanish. And I knew that I was good in chemistry and Spanish, but I didn't know if I was gonna be good in chemistry and English. And I just thought, I'll figure it out. And I ended up going, and yeah, it was tough, but I made it and I had a lot of support, and I wanted to talk to you about this. So the summer before I went and started school, I got an invitation from this program, Student Support Services, and they told me, oh, you are eligible to like come early. And I think it was like a 2 or 3 week program where I would go to campus and nobody else had moved in. So I would go to campus and we stayed there, and it was a great chance to build community with people that were like me. So this program is a federally funded program to support students from low income or first generation or students with disabilities to finish their education after high school. And at the time, I just saw it as like a fun opportunity to go there early and explore and meet people. And it was really useful because I met a lot of my friends that ended up taking classes with me and we would study together. It definitely bonded us in those 2 weeks. And then we were sort of connected throughout the rest of my undergrad experience. So I always felt like I had people that I could connect with. And additionally, they had things like advisors that would give us advice when we were choosing classes and everything, and we didn't have to wait for the long times for advising at the university provided counselors. So it was very useful. So through that program, I was doing well in my classes, but I was definitely not getting straight A's like I was used to. And this was, of course, like the language was different. The classes were super hard. A lot of people, like I was overwhelmed at certain points. And I knew that if I wanted to go to medical school, it was going to be really competitive. And I had good grades, but not perfect grades. So I was thinking, what are ways that I can boost up my resume so that when I apply to medical school, I can get in? And I thought that research would be a good opportunity to boost up my experiences and become a more well-rounded applicant. And so through the Student Support Services program, they told me there's this other program that's called LSAM, that's the acronym, and it's an NSF funded program to support underrepresented minorities in STEM. And so I joined this program, and they funded my first summer as a researcher on campus. So I got paired up with Dr. Jeff Matebesky's lab, which was in the physics department. And I studied the synthesis of nanowires to include in flexible organic electronics. So the fancy LEDs and OLEDs and all of that, those are based from these materials. And that was like my first entry point to me doing an experiment, me figuring out what materials I need to do, what are the controls that I need to do. And I really enjoyed the experience. I thought it was awesome that I was getting paid to learn. And that actually ended up giving me the chance to present like the first time at research conferences. So I went to 2 research conferences to present the work that I did that summer, one in Albuquerque and one in Niagara Falls. And at that point, I thought, wow, this is crazy that somebody is like spending all this money on me. And all I have to do is like show up and learn about things that I don't know about. And the other part is like, they're paying me to go to places that I never would see if I wasn't part of this conference. And once you're at a conference, yes, you're in a conference, but you're free to walk outside and like visit other places. So I really enjoy that part of it. So then my next summer, I decided that I wanted to branch out a little more. And so now I didn't want to stay in Binghamton because in the summer it's like a little dead because a lot of the students go away. And so there was just like a few of us that were doing research. So I was like, I just want to learn about a new place. So I applied to this program, which was also funded by the NSF, but I got paired up at University of Illinois in Urbana-Champaign. And there I went back to a chemistry lab. So here I learned about gold nanoparticles, how to synthesize them, how to characterize their size, their charge. I learned that you can load things onto these gold nanoparticles and deliver them to certain parts of the body to deliver a drug perhaps. So I really started to enjoy research more because I saw more of a connection to like human health. And so I realized, you know, I'm giving all these experiences, and prior to this, my only experience was in a physics lab. And it didn't matter that that experience had nothing to do with this one in terms of the content. It was the skills that I was acquiring through these experiences that were making me a better applicant for these opportunities. So here, because it was through the Center of Nanotechnology or Sustainable Nanotechnology, I was in University of Illinois, but there were a lot of other students that were at University of Wisconsin or like University of Maryland, Johns Hopkins. And at the end of the summer, we all got together and presented at University of Wisconsin. And I went there in the summer and it was awesome. It was beautiful. And it was a great opportunity just to see other people. And this was actually the first time that I ran into another girl that was from Puerto Rico. And she and I became like really good friends. And it was just like a very exciting time. And this was the summer of my junior year in college. That same summer, I was given the opportunity to go into a science exchange program in Germany. So this was a new program that the German government had sponsored. And what they wanted to do was to establish a program in which students from Binghamton could go to universities in Germany and German students could go to Binghamton and foster collaboration across the countries. And we were like the first group to be sent as ambassadors to show what was going on at the university. So I had my research project in that physics lab. So I applied and then I got approved to go. And everything was paid for, so science sponsored my first trip to Europe, which was awesome. And it was a 2-week trip, very intense. We went to like 5 different cities, and you can see that they're scattered like all throughout the country. So we took a lot of trains. It was very intense. We went to Berlin, Dresden. I'll show you a couple of pictures. But again, it was this idea of you're sharing your knowledge and your research across the world. You're learning about what other people are doing, and you're also getting to explore places that I didn't have the money to take a 2-week vacation to go to Germany. So it was very fun. And these are like some pictures of that trip. And in the first picture, we were just walking around the town, and the person in the center of the table is the head of the chemistry department, which was crazy that I was just like drinking Riesling with him in the middle of Germany. But it was really fun, and it was a good time to see that even people that are the head of a department are human just like us, and they can like sit down with you and talk to you about science, of course, but they can also talk to you about like their families and their experiences. And you don't have to be this perfect, all-knowing being to be a scientist. And actually, to be a scientist is to recognize that you don't know a lot of things and trying to find the answers. So anyway, that was a really good trip. Got to see a lot of fun stuff. And then in the fall, when I came back from this trip, I was supposed to be studying for the MCAT and like applying to medical school. And I was not feeling it. It was super stressful. I had like, I don't know how many breakdowns of, oh my God, what am I putting myself through? This is awful. And then I just remembered, I just had this amazing summer with all of these experiences. And why do I want to go to medical school, really? And it was just this idea that I had had in my head since I was very little. And it wasn't necessarily that I was even passionate about it at that point. Like, I didn't necessarily see myself interacting with patients directly. It was more about the curiosity of the, like the disease, how the body works. Like, that was the area that was attractive to me. And yeah, so I remember, like, they're paying me to learn. Why do I have to go to medical school and get myself into a ton of debt where people can pay me to learn and get a degree and do the things that I've already been doing and that I've enjoyed so far? Like, there's no point of me sitting here crying over the MCAT. So I decided to apply to grad school. And again, I think I was a strong applicant to grad school, not because I had perfect grades or because I had any number of publications. At this point, I had had all of these research experiences, but I still hadn't had a paper. And it's very unusual for undergrads to have papers published, but some people do. And when you get to grad school, definitely you hear people saying like, oh, I have 3 papers already. And you're like, okay, I don't, but I'm here with you. So I think, there's this idea, and I've been guilty of it, of thinking, well, I don't check all these boxes, so there's no way that I'm going to be the applicant that they want. There's no way that they're gonna admit me. But what I did have was a lot of experiences. When you're applying to grad school, I think they're looking for you to have some sort of idea of what to expect, because grad school is very intense. And the fact that I had done research at different places meant that I had talked to a lot of PhD students, a lot of postdocs, and I did not experience the stress that they had, but I did see kind of like what their day-to-day was. And I had started to learn skills, how to think critically, how to set up experiments, how to ask for help. And additionally, I had letters of recommendation that I never read, but they were probably good because I got into grad school. So I ended up going to UVA, University of Virginia, because again, I was in Binghamton, that was in upstate New York. And I'm from Puerto Rico. I wanted to get away from like the really harsh winters. So I was looking for somewhere that one would have projects that excited me, but also that didn't have crazy winters. And Virginia gets a little cold, but not that bad. And I knew I wanted to work on something really related to human health, but I didn't know necessarily what that was. I knew that I had seen nanoparticles being used to deliver drugs, or they were being made with that goal in mind, but I didn't know if I wanted to necessarily do nanoparticles. So what I did when I was applying to grad school was read on the faculty and see if I could identify multiple people that I was interested in, and then go to that school, shadow, go to their labs, and then figure out like what clicked with me the most. So So in graduate school, I ended up working in integrating hydrogels with microfluidics. But before I get into all of that, I wanted to take a step back and talk about a concept that you probably learned in maybe high school biology or middle school, and it's homeostasis. And this just means that our body is made up of organs that are connected to one another through the vascular system, through the nervous system, and all of these organs are communicating with each other, sending signals to one another. And when everything is working as is intended and harmoniously, that's when we're healthy. Whenever there's a disruption to this perfect harmony, that's when disease begins. So for example, in cancer, if you think of someone that has brain cancer, there's all these cells that are invading the space where they're not meant to be. So in order for us to develop ways to treat certain diseases, it's important to recognize how the communication works when we're healthy. Because that way, if we understand that, we can, design drugs to deliver to certain tissues, or we can more easily design things that are meant to bring back the system to harmony. And then the other thing is that, like, as I mentioned, we're made of organs and tissues, but there's also a lot of flow in our body. We have like lymph fluid, we have blood that is applying some pressure onto these tissues. So it's not only the fact that we have organs, like those organs are connected through fluid. So the lab that I ended up joining was using microfluidics to understand immunology. And this was the first time that I learned about microfluidics. I had never seen it before, even though, I had done all this research. I think microfluidics is usually kept on the engineering side of things, even though a lot of the platforms that are developed are meant to be used by chemists, biologists, but there is still a gap of translation. So when I saw microfluidics, I thought it was so cool that you could make something up. I guess for those who don't know, microfluidics is just a device that you can hold in your hand, or maybe you can hold a couple of them in your hand. They're very small, and you can imagine it's like a plumbing system that you can shrink. So then you can create reactions and really fun science because of that length scale that is really small. So like the channels are the width of like a couple of hairs put together. And then you can connect those devices via tubing and syringes to whatever fluid. So you can imagine, in the analogy of like homeostasis and your tissues and your body, you could have cells inside of these channels or chambers, and then use the tubing as the vasculature in your body. So at the time when I joined the lab, my PI had just began, and I was only the second generation of students that she had taken in. And she had a really big interest in lymph nodes. And for those who don't know, lymph nodes are vital for immunity. So a human has between 500 or 600 of them all across your body. And they're scattered throughout your body because they house the cells that fight any antigen or foreign substance that is attacking your body. So if you only had lymph nodes in your arm and then you have something attacking in your leg, the time that the cells would have to go to travel and be able to respond would be too long. So we have we have a lot of them so that we're all protected, and the time that it takes for a cell to get to the point where they need to fight it, they don't have to like crawl all across your body. So that's why we have a lot of them. And the other thing about lymph nodes is that they have a very specific architecture. So when you look at an organ, you'll see the regions. For example, the picture at the bottom is a slice of a lymph node from a mouse, and they're stained with specific antibodies to highlight particular cells. So for example, you could see that there's like circular regions that contain B cells, and these are the cells that produce antibodies. Then the dendritic cells are labeled in green. And so for example, what happens when a foreign substance enters your body is that a dendritic cell would recognize that substance and then would I was gonna say fly, would go through your vasculature, enter your lymph node, find another set of cells that are called T cells, and tell it, hey, look what I found. Can you prepare to attack it? And what happens is that T cell gets activated and starts to replicate, and you start to form like an army against that infection. So whenever you're getting sick and people say like, oh, my tonsils are inflamed or my lymph nodes are swollen, that's because your lymph node is expanding to be able to sustain all these cells that are getting ready to attack whatever foreign substance. And then when they leave to go and fight it, that's when they go back to normal. So it's interesting that there's this very specific architecture, and whenever you look at the organ, they're always, the cell types are kind of sequestered to specific regions. And so we really wanted to understand like, what about this spatial distribution facilitates the immune response? What makes it effective, and why did it evolve to be that way? So at the time that I joined the lab, my PI had developed this model of tissue slices as a way to investigate these questions about spatial organization. And in this picture, you can see these like blue circles, those are agarose gels, and inside them, there's lymph nodes that we take from mouse, and you embed them in this gel. And because it's clear, it's easy to image and take pictures like the one that I just showed you. But once you have this block of gel, it's probably like this tall, you can take a really fancy slicer that's called a vibrotome and slice those into very thin slices. So from one lymph node, you can get multiple slices. And what's interesting is that even within a lymph node, those are pictures from the same one. And you can see that the cells are kind of distributed in different regions, like as you go in deep into the tissue. So if you want to do a mechanistic study of what happens when cells are 200 microns apart from each other versus what happens when they're really close together, using the tissue slices makes that complicated because you have no idea what organization you're going to get when you're slicing these. So we really needed a way that we could kind of create the organization from the bottom up and then be able to ask those questions. And so the techniques that were available is using in vitro cell culture, which that just means instead of slicing the lymph nodes, you take the whole lymph node, you crush it, you pass it through a filter, and then you suspend it in some buffer with some nutrients so that the cells are still alive. And in that type of model, it's very easy to say, for example, okay, let's just take a pipette and put X amount of a drug onto these cells and they'll all receive it. The other part is in vivo experiments where instead of adding it into a dish, you can just inject an animal with that drug, and then you would have to sacrifice the animal and then see what happens inside. That's difficult because you're not seeing things as they're happening in real time. You're just seeing the endpoint. And so it's hard to understand the dynamics of what's going on when the drug enters the lymph node, for example, and track it over time. So we really needed an intermediate approach that could bridge those together. And another thing is that, for example, when you crush the cells, you lose all the spatial organization, and you also lose the support of the tissue that's holding the cells in place in the organ. And if you read a lot of papers, you'll see that this is very important and that the force exerted by the tissue also affects the way that cells respond. So then that's where like the goal of my thesis came. And it's like, okay, can we make a small functional unit that we can manipulate to replicate the organization of these cells in a very direct manner? And then if we put those cells inside of a chip, then we can also include fluid flow to see how that affects the function. So it was quite the big goal. And on top of that, as I said, she had developed the slice model. She had never worked with any like biomaterials. So I was also in charge of kind of like starting this new wing that the lab was going to explore. And I thought at the time, this is awesome because like once it's going, you know, I know that I made this and I'm gonna leave a legacy. And like that is true, but it's also so much work. I think usually when you join a lab, especially if it's not a newer faculty member, you're kind of picking up on the progress that somebody else left. So you're not starting from zero. And sometimes, of course, that comes with challenges and like, it's still hard to get a PhD that way. But you perhaps have a few more wins really quickly that kind of push you through. Whereas for me, this was very intense because I kind of had to figure out everything. Like even from what material are we gonna use? What is the best one? And I'll go through the other slides and tell you like the decisions that I had to make. But at the time, I thought that it would be really cool to use what it's called photoreactive hydrogels. So in this, with this materials, you start with a mixture of a macromer, which is normally a protein, and in some cases the linker, or sometimes macromers react with each other. And then you have a photoinitiator and a wavelength of some, a light of some wavelength. And I didn't know what photoinitiator to use. I didn't know what macromer to use. I didn't know what concentrations to use. So all of these were things that I had to figure out. So like my first years is a lot, well, I would say, yeah, like my first year was a lot of planning, reading, learning what were the methods available and kind of like choosing what would be the most promising way, and then testing a lot of stuff that didn't work until you hit something that works. So you have all of this, and then in the presence of the light becomes a hydrogel because the proteins, the groups in the proteins react together and form a matrix. So I decided to use gelatin as an extracellular matrix, and extracellular matrix just refers to like what builds up the tissue in your organs. And so gelatin is just denatured collagen. So collagen is one of the most prominent proteins in your body, and it's about a third of the total proteins that you have in your body. So I knew, okay, at least this is not going to be toxic because it exists in the body. And the other thing is that it retains bioactivity. So for example, there are places in the gelatin that cells can interact with, and they can break down the protein to kind of make space to move around. So I knew, for example, if I wanted to see in lifetime how one cell could come in and then tell the T cell, I recognize a foreign substance. That cell has to like crawl through the space to find that other cell. So I needed something that could be reactive to the cells. So I thought gelatin is good. The only problem is that it's not very stable at physiological temperatures. So people have had some tricks. You can add pendant groups or functional groups onto the gelatin backbone, and those groups then can be photoreactive. So in the presence of a photoinitiator, for example, the methacrylate group, wish I had a pointer, that group could react with another group and build these covalent bonds that kind of create like a mesh. And so you can have cells within this matrix. One thing that was important, as I mentioned, is that in diseased tissue has a different mechanical property. So sometimes, for example, like if you have a tumor, it could be a lot stiffer than if you have a healthy tissue. And those forces also impact how your cells behave. So first thing, or one of the first things that I had to do was figure out how can I match the stiffness of these hydrogels that I'm making to be similar to the tissues of the lymph node in the body. And for that, we use a tool that's called a rheometer, and it's a stage that has a piston. And at the bottom where you put the gel, you can shine the light that catalyzes the reaction, and you can track the stiffness because the piston is putting like a shear stress onto your matrix, and you can link the resistance to that deformation to the stiffness of the tissue. So I did a lot of these experiments where different formulations of different chemistries, different amounts of photoinitiator, because you want to control the time that you're exposing the cells and all of that to try to figure out the correct one. And that was like a very long story because also I was the one that was functionalizing the gelatin. by the end of my PhD, you could buy it, but I had to make it for the longest time. And there was a lot of issues with irreproducibility and things like that. So long story short, I was able to figure out how to match that stiffness. And okay, we have the stiffness done, but now are these gels stable? And that was the next thing, and the next big problem is that the stiffness matches great. Now, when we put it on the chip, is it going to be there once we start flowing all these media that is like mimicking your blood flow, for example? So the idea that I had is that you have this precursor that contains your macromer, photoinitiator, you flow it inside of your chip, and then you can have a photomask that has whatever design you want to create. So let's say that I wanted to have one spot that represented the B cells. So I would have a mask that would have a transparent center, and then I would flip it upside down and then shine with the light, which would cause the gel to just be created in that area that the photomask allowed the light to pass through. And then I would rinse out the rest and then be left with whatever design I wanted. So this is what the patterns look like. So at the top is the design that I created on a photomask, and then at the bottom is like the actual gel on the chamber that has already been cross-linked. And this is nice because you can be iterative with it. You can have one mask and one design, or you can do one and then surround it with a different set of cells, maybe a different gel that has a different stiffness to it. And you can continue to do these several times. And you can even make like fun patterns. Here's like the famous rotunda that's very famous at UVA. And then, so now I had figured out what chemistry to use, how to make these patterns, how to have them be stable under flow. And now it was time to add cells. And so when I first started the project, everything was using mouse cells. But because eventually what we want to do is be able to have an organ that can represent like the cells in your body, like take a patient's cells, put it on the chip and see how it reacts to a drug, for example. We wanted to move towards a human source cells. And so we got this big grant that allowed us to make that transition. And what's cool is that when you go to donate blood, like platelets, for example, when they take the blood, they're only interested in the blood that in the cells that clot and prevent you from like bleeding during a surgery or during an organ transplant or something like that. And the rest of the cells that are the white blood cells are discarded. Or in their case, they can also sell it to us and then we can use it in our experiments. So we started getting all these human cells and then we were able to start putting it on the chip. And this was like really good because they were actually more hardy or stronger than the mouse cells. So my experiments and like the viability of the cells started to become a lot better once we made the move to human. But the problem was there was a lot of delays during the pandemic as well because people stopped donating blood, like they weren't going in the same amount of numbers. So sometimes, and the other part is that these cells would come in the morning, then they had to be processed by our collaborators. I would only get these cells at like 8 p.m. And the rest of the day I had been prepping to like run an experiment. And I just had to sit there until 8, until the cells came in to start running these experiments. And there would be days where I did all of the prep the whole day. You know, it was like, I don't know, like a 12 or 14-hour day. And then they were like, oh, the cells didn't show up. So it was like really a crazy time. And, you know, this is just like one example of things that are totally out of your control. And you have to kind of figure out what to do next. And we were able to make it work. And these are like examples of patterns that I did with cells. So in this case, my favorite one is like that alien. You can't tell. Yeah, it's like an alien face. I just had a lot of fun like with the designs that I did. So anyway, so I can pattern cells, they're stable. And now I have to culture them. And this is a picture of the incubator. Oh my God, I wish you could see from my screen maybe. But it's a lot of cables. So like the devices are tiny, but everything that drives the flow, it's always really bulky. There's a lot of connections. I had to create like these little holders for the devices, little connectors that would make the tubing stay put. Because the worst part was when you would spend like 15 hours setting up an experiment and the next day you come and the cable was like flopping around. That happened a couple of times. So you also have to, it's just constant problem solving after problem solving. So there's a lot that you learn in grad school. So I'll go a little bit quick, but the idea is that I was able to create the system that you could put cells in any sort of design that you wanted, and the cells lived. And then because of that, we got a grant that extended that technique onto another laboratory at Virginia Tech. And now they're currently doing amazing things. And when I graduated, that paper of this method that I developed was under review. And I had only published another paper with another student that was also in my lab. So there was a lot of stress of like, wow, I only have one publication in this paper in review, and I'm supposed to be looking for jobs. And, you know, I had a moment where all I could think is like, all the things that I wish that were in my resume that weren't there. And that's a really toxic mentality, because there was so much that I had learned. There was so much that was in my resume that I just couldn't even see, because I didn't have this one line of the paper. And it wasn't lack of work. The paper was written already, you know, it's just like waiting on these. So I think I also learned at that time that you gotta trust yourself, and that when you go into interviews, you will have things to talk about because you've done the work. And you can't compare yourself to others, because it's very easy to say like, oh, this person had another project and they got like 2 papers by the time they were in their 3rd year. And, you know, this is something that happens. And I'm trying to find ways of how can I make this environment like healthier for us, and so that we don't always feel this need of compare ourselves to others. So in that note, like grad school, I did a lot of stuff beyond the lab as well. I was part of the biotech training program. This was really cool because they funded part of my PhD as well. So my first year I had to TA a class, and that definitely limits the time that you have in the lab. So once you got extra funding, then they can pay for that and you don't have to work for the university anymore, and you can just focus on your research. We went to tour like the FDA and other companies, also a lot of practice presenting the research. They also funded an externship where I worked for a local company that worked on centrifugal microfluidics. And these are different from like all these tubings and connections that I showed you before that I had to deal with. These are all in like a CD type of format, and they have channels that are cut by lasers. And the way that you move fluid is you put this disc onto like a CD player, and it just spins really quickly. And because of centrifugal forces, like the fluid always moves outwards, and that's how you move fluid through. So there's like layers that would block fluid, and then you can take a laser, melt it, spin the disc, and move fluid. So that was like a really cool experience of microfluidics in a totally different setup. And I also went to a lot of conferences. I was very lucky. So I went to Phoenix, Arizona. I always try to find like at least one thing to go and explore whenever I'm in a city, because they're paying for my hotel, they're paying for my meals. Usually you get reimbursed. So it's like, I can probably afford a $50 Uber to go and see what I want to do. So I'm definitely like a huge fan of going to conferences. I went to Seattle for another conference, and I was able to see some of my friends that live there and see the cherry blossoms. And then I also went to Hong Kong. Like, I never thought I would go to Hong Kong. And it was the best conference. This is actually where I met my current boss. And the way that I met was because she was giving a talk, and in the corner, she had the picture of the grad student that had done the work. And I saw that they had like a Latinx last name. And I went up to her, I was like, this is the first time that I've seen someone with like a Latino like last name on any microfluidics presentation. Like, that's awesome. Can you tell me about her? And that's how we met. And then later on, when I was looking for jobs, my advisor sent me the posting from this person. I recognized her name, and I said, oh, I met her and we talked. So I emailed her. And she remembered me, and that's like basically how I got my job. So I think a few reflections on grad school is that it gave me a lot of experiences. I became very resourceful, very good problem solver. I learned a lot, and I'm proud to have finished because it was hard. There were a lot of times that I was like, I don't know if this is worth it. Like maybe I can just take the master's and not finish. And I thought back of the experiences that I had during my undergrad and why grad school was so much more difficult than anything else I had done before. And of course, the rigor of graduate school is very intense, but also I felt very alone. Like there weren't a lot of people that looked like me in my department. I was probably the only Latinx student in the department. At the time I was going through like issues. For example, there was like this big hurricane in Puerto Rico, and I went like 6 days without hearing from my parents. And I didn't have anyone around me that could relate to that. And I realized like there's other people like me that are the only ones in their departments that are feeling really alone. So when I graduated, I was like, how can we make academia better for Latinx people like me or other underrepresented students? Because these are issues that, students that come from backgrounds that are more represented, they have role models that they can look up to, that they can see, okay, they overcame adversities that are similar to mine. It's easier to connect with them. And this is not only me, right? Like there's 19% of the US population is Hispanic, but only like 7% of the engineering degrees go towards people from Hispanic or Latinx backgrounds. And yeah, like I said, like usually you don't have role models. So I'm really trying to increase representation of all sorts of people that are not represented usually, so that they can then be in a position where they can inspire others, where they can have like honest conversations of the hardships that they've gone through and the things that they've overcome. So I think the work that I'm doing now is doing that. And I think I wanted to still apply like all the technical knowledge that I had, but I didn't want to be doing experiments because it seemed like there's a much bigger problem that we should be addressing. So I think like right now I'm doing the best of both worlds. So I'm gonna skip ahead a little bit, but essentially the Foundry is a clean room space that is shared with people across campus. And it's a very clean space because again, you're making tiny things. So if a clump of cells falls onto whatever you're making, it's ruined. So it's a very clean space. And this is like the class. I can talk to you more about that if needed, but activities that take place there. Train and research for graduate students and postdocs. And particularly people that are not experts in microfluidics. Like that's what I love when somebody comes and says, oh, I have this idea, like, can you help me figure it out? And then I act sort of like as a consultant. And if somebody comes with something that I know it's not gonna work because either I've seen it not work before or it's too complicated, or if I have a trick that can help them, that's what I do. And then teaching, so I help teach a class during the winter quarter and educational outreach, and then development activities that are essentially like fundraising. So I've trained around 18 users since I started last year from a bunch of different departments on campus. And it's a great time. Like I just really enjoy seeing people like the first time that they make something and they're like, wow, I made this, it's crazy. Yeah, so quickly about the outreach. And I think we started like 5 minutes late, so I can have like a little bit more time. So the microfluidics device lab, this is us gowned up to prevent, it's actually to prevent the space to be contaminated by our cells and not protecting ourselves from the environment. So we teach this class and 20 students, and the goal is to pair up graduate students from bioengineering with life scientists across campus to have interdisciplinary collaboration. And you need collaboration because every new animal or cell that you want to put inside of a device, it's really hard to predict how they're going to behave. You might have to coat your surface differently. It might be that somebody wrote a paper about encapsulating the same animal or the same organism that you want to. Let's say it's like, I don't know, a bacteria or something, or maybe a worm, and they posted this online, and then you try it and you realize, oh, the ones that I'm using are slightly smaller, and that means that you have to redesign the entire device. So there's a lot of troubleshooting and like redesigning and iteration that has to occur. And for somebody that, their expertise might be more on like genetics or things like that, they don't really have the time to focus on learning this whole subfield. So pairing up with bioengineering students creates like a perfect environment where the bioengineer student is kind of troubleshooting for them. And at the end of the class, or usually at the end of the class, there's a prototype. So the way that the class works is that the labs come and they pitch essentially to the students, like, please take my project. This is why it's cool. This is why you should work on it. And then the students work in teams and they choose the projects and then they design and fabricate them. So we teach them design rules, how to fabricate. We teach them how to test them. And then because we know that science like very rarely works the first time, they have a chance to iterate and redesign and retest. And it's amazing the difference or the improvement between the first round and the second round. And I always tell Polly, who teaches the class, if they had another quarter or even like another month, this could be something that would be ready to be published. Not all of them, of course, but maybe like 2 out of the 5 projects that we do are like really at the cusp of, being totally done and to be adopted by another lab as well. So for example, some of you might have seen these, but I just love these videos of anemones trapped inside these arrays. And these were used to understand coral bleaching and the symbiotic relationship between algae and anemone. So the anemone provides inorganic nutrients to the algae, and the algae provides the glucose or other carbon-based nutrients to the anemone. And under stressors, what happens is the anemone doesn't want to hold on to the algae anymore and kind of like vomits it out. And so then that relationship, it's broken down, and that's what we know as coral bleaching. So you're seeing it in that video right there. Yeah. So that's an example of something that was in the class and was actually published. And this one, the other one that is a little intense, is the flatworm being bisected. And so you can imagine you have like this little worm that it's like really small and it's also like moving around. How do you, with like a razor blade, like you couldn't be able to dissect it. And they're important because they're really good at regeneration. Like they're known for that. So flatworms are a model that people used to study tissue regeneration or tissue regeneration for things like you have an accident and like a patch of your skin needs to be replaced and things like that. So that's why. And so it's dissected, and then each half is collected and cultured, and you can see that they're like little babies, but they'll grow, but they're moving and they're alive and have eyes, which is really cool. So at the end of the course, they have these working prototypes, which is super exciting, but usually they have to go back to their own research. And the way that the collaboration works in the class is that they'll test the devices, and then their collaborator lab would say, okay, let's try this experiment, or this is the type of media that you need to use and things like that. But there's not a lot of time for the students to teach the lab how to use the device. And they have their own research to get to. So I think the cool thing about these projects is like they're vetted, right? Whoever takes over these projects, it's not like me trying to start a whole project from zero when I was doing my PhD. These are projects that have a really solid foundation, and that somebody else, you just need like a new set of hands that can continue that transition into the lab. So how do we leverage the successes of this class while providing opportunities for a new generation of students to learn and test and do research. And this is what the motivation for like the outreach programming was. It's like, this is a perfect opportunity for somebody that even doesn't have any experience in research to become familiar. So we have raised some money for summer internships where any of you could apply and come and do research for 8 to 10 weeks on campus. You would receive a mentored experience where like me and other people from the Foundry would teach you how to fabricate these devices, how to troubleshoot, how to test them. And then you would also have creating like a network with the people that are also going to be in the program, but also with the people that are mentoring you, with the faculty that are mentoring you. And I think the other important thing is that, you know, we right now have money for anywhere between like 4 or 6 students for next summer. But the idea is that there are other programs on campus that are offering opportunities for students like you. And we've been talking very strongly about coming together so that you have like a bigger cohort of students where you can talk to each other, and maybe you do the research, and like me with the physics lab, they just don't manage, but you're getting experiences and you can talk to your friends about what they're doing, and maybe you can stop by and like see what they're doing in the lab one day. So this is just like a paid opportunity where you're gonna learn skills that regardless of whether you want to go into grad school or if you want to go and work in industry, you're gonna use these skills for those jobs. So I'm really excited, and you know, if any of you have questions about like what type of projects or you know, like what the experience would be like we can talk about that too. And then because that only happens in the summer, that's why I created the workshops, like the ones that some of you attended in the summer. And that actually was like a very short version of what I envision, because I want to be able to show you like how to make your own device. And maybe we can run an experiment, but you can learn how to make it and how to do a demonstration, things like that. So these are just pictures, and some of you might be on them, of the workshop that we did this summer. And we were so excited. I was so excited to host you. And I just had the best time talking to you afterwards and like hearing all your ideas. And like some of you told me like, oh, I don't really know if I want to do this, but I'm really interested in medical school. And then I got to bond with you over like the stress of that. So I think I just love being able to talk to people and just realizing like, I have a PhD and it was hard, but at one point I was just like you, and like, we are here to support you. So yeah, just pictures from that trip and all the fun stuff that we did. That's the setup with all the syringe pumps and some of you admiring the microfluidic devices. So yeah, so I guess like I just wanted to finish the presentation with like a few points that I've learned along the way and that I hope that come through the presentation. It's like there's so many things that you can do in STEM, and you don't have to stick to the first one that you try. Or if you don't like the first one that you try, doesn't mean that you're not gonna like the next thing that you try. So I think the key point is like, if you're interested, explore and explore a lot. And even if you like the first one that you try, just try another one and see if you like it better. A career in STEM is achievable, and there are people out there like me and Sophia that wanna support you and try to help you find these opportunities, because there were people out there that suggested me these opportunities, and that's the whole reason why I did all of them. I think when you're in school, it can be overwhelming to think beyond the classes that you're taking and things like that. So people that like, it's our job to try to match you with opportunities. So use us. And yeah, I think again, the lessons learned through the research will translate to jobs outside of academia or within academia. And talk to people, because you never know what the future holds. When I talked to my current boss at that conference, I just talked to her because of that small picture that I saw in the corner. And I didn't imagine that I would ever be at Stanford working with her. So it's just crazy how the world works. So if you see someone that, I don't know, but for any reason seems interesting, just reach out and talk to them. And I know, by the way, I know that's hard to say because I'm really shy. Like, it's really hard for me to like go up and talk to someone. So, and again, I did it a few times during my whole graduate career, and it ended up working out for me. So it's like, just put yourself out there. And yeah, most people want to help you. and we can make STEM better, but we need you, so come. And yeah, finally acknowledgements of like the main groups from my PhD and my current lab and all the people that have helped me along the way. And if you have questions, I would love to answer.
Well, let's give Dr. I love the personal as tied to the research. So let's see if anybody in the room has questions, and we'll also check if there are questions in Zoom. Yeah, anybody in the room have any questions you wanted to ask Dr. Cardenas?
Yeah, I'm having a little trouble kind of like putting it together, but I remember when I went through, I kind of imagined that it seemed very bio-focused, the lab. However, then seeing the other grad students you had, they were from all over the place. So like, for example, with electrical engineering, how does that go fit into?
Yeah, so the person that, I think it's a little bit bio-tied. So for example, the person that is in the lab that's doing electrical engineering, they're doing, they're incorporating electrodes onto the microfluidic devices to send stimulants to the cells and then see how the cells react to an electric field. So yeah, but I think the microfabrication, especially like nanofabrication, can be more in the, what's the word, like material development and things like that, that might be more like electrical engineering focused. But I think what we do at the Foundry, it's always trying to tie together like hard science with like life science.
Thank you.
While people are thinking, I'd love to hear, I mean, you've talked about your journey and it sounds like it was a lot of trial and error and that kind of thing, but what kind of people were there along the way that helped keep you in it? Because it sounds like it was also very isolating, being in that field.
Yeah, I think, and I've been learning this along the way, is that, and that's why I said it at the end of the slides, like talk to people, people want to help you, because I feel a lot of times, you know, you want to put on this facade of like, yeah, I know what I'm doing and I can do it. And sometimes it takes for you to get really low to then be like, I have to ask for help, because if I don't ask for help, like I'm not getting out of here. And so I had people, like once I opened up to my advisor, she was very, you know, adamant of like helping me feel more connected, and she sent me to like the offices of like diversity and inclusion. So there were people at those offices that connected me to other students that maybe were years ahead of me. And then I had an outlet to talk to people that had gone through the same thing. But when you're feeling bad, you're not Googling like diversity and inclusion office, right? Like that didn't even cross my mind. So it took like speaking up, and you know, you have to find people that you're able to trust. But I think, you know, from where I started to where academia is now, I think these issues are a lot more outspoken. So I think there's a lot of people that, and especially departments, are hiring a lot of people that are meant to support people that are going through these struggles. So.
Anyone else have any questions?
I did have another
Yeah, no,
one.
go ahead.
Sorry. Earlier you mentioned that you touched on the rigor of graduate school. When I was watching the presentation, I was surprised how hands-on it looked, how hands-on it seemed. When you mention rigor, are we still also talking like academic rigor, like things to be learned, things to be researched more into for understanding? Or is it a combination of the hands-on, the things you have to come up with, and the academic rigor?
Yeah, so my experience, and I think different programs have different requirements for the amount of classes that you need to take and things like that. But for me, the first year was all taking classes and then kind of shadowing in the lab what people were doing, kind of coming up with the idea of what the project I wanted to work on was. I think the classes in general, and especially because you're taking these classes with a bunch of people that just joined the program, you find a lot of community in that first year to get through the classes, because they are difficult, but if you got into grad school, people want you to succeed. So the classes are not meant to weed you out like in undergrad when you're in a giant classroom with 300 people, you know, and you feel like you're just another face in a sea of faces. I think a lot of things are more personal in graduate school. So the only class that I found really difficult was like a coding class that I took. That was awful, and I don't know why it didn't connect with me. But other than that, it was doable. I think the rigor comes from you are in charge of your project. And if you don't know something, nobody's gonna come up with a perfect solution for your problem. They might suggest things, but at the end of the day, it takes trying and trying and trying and getting really used to failure. I think it's probably unnatural for humans to experience that much failure, but what you need to think about.
Oh my gosh.
No, but I think you're failing, but then you're also learning. You're learning how to better design your experiments. You're getting better at how to discern what's a good idea and another idea that might not be so good.
Yeah.
So you are learning, and the good thing is that you become a source of knowledge for the people that come after you. So for example, like when new people came into the lab, I had gone through a lot of mistakes, and I was able to give them like advice on like, these are the protocols that you need to follow if you want your things to work. And they will eventually run into their own problems and learn how to solve them. And so like, that's why I say that being a scientist, like you're just learning that it's like the deeper you go, the less you know in a general sense, but the better you get at solving your particular problems. So yeah, I think getting comfortable with failing. And I think whenever, it's hard to say those things out loud, but whenever you enter a room of graduate students, like they'll all be saying like, oh, my thing didn't work. And then when you go to a conference, all you see are the positive results, right? So that's another issue with like the way that we, I guess, promote science, where all that gets published are the things that worked, and all these data of mistakes gets just kind of hidden. And the only way to learn is like by talking to people. So.
Thank you.
Any other questions? Any questions in the chat? No. Well, thank you so much for your time, Jennifer. It was wonderful to hear your story and learn more about your research, because we did get to go to the field trip, and we are hoping to do more field trips in the coming year as well. So let's give Jennifer a hand once more. Just a couple last announcements. Do you want to just switch over to the SLI slides? Our next talk is in 2 weeks, so it's totally different discipline, astronomy. Dr. Sophia Sheikh from the SETI Institute will be coming, and it's a different time also. So it'll be Friday, November 4th from 11 to 12:30, and we'll have lunch afterwards. It'll be burritos next time. So please come and join that same room or on Zoom. And then the last slide, if you all could, for those of you who joined us today, if you could fill out our exit survey. There's one more slide, really? Yeah, could you all just do our exit survey? If you want to do it on paper, Fatima has paper, but it's just a few questions to get your feedback on today. It's an anonymous survey. Yeah, Alex, do you mind doing that too? Just
I can't believe this works.
yeah. It should work because it's huge.
Yeah, it worked.
Go ahead, Manisa.
If students want to apply for those internships that you talked about, like how would they do that, and where would they get information about that?
Yeah, so the applications aren't open still, but I'm in communication with Sophia about this like all the time. So when they're open, I'll definitely share with Sophia, and then.
Yeah, and then if you are on our email list, then you will get it.
Yeah.
And if you're not, you should join our email list.
And also you have my email, and I think
Yeah.
I agree.
Yeah,
to
if
have the
anybody-
email
yeah.
out, so.
Definitely. If anybody
If you-
wants to contact Jennifer, please let me know. I can contact you.
Yeah.
Link you up with them.
Yeah, and the other thing is I mentioned there are other programs on campus. So like if you, for any reason, feel like microfluidics is not what you would like to try, I can also put you in touch with other people that are doing other type of things. We're really trying to, one of the things that we always talk about is that there needs to be a centralized space. So we haven't built that, and we want to build it, but in the meantime, like I can connect you with them. So feel free to reach out.
Well, feel free to come up to Dr. Cardenas to talk to her a little bit more. Thank you all on Zoom for showing up for us today. And we're going to sign you all off. And we hope to see you all next time. Thank you.